organ we're going to look at that's controlled by the pituitary gland is the thyroid. And you saw this in the models yesterday during lab. So the thyroid gland is that H-shaped organ that is found anterior and then lateral. So the larger, taller components of the thyroid gland are found on the lateral aspect of the trachea. In fact, if someone has a tracheotomy, maybe they need it induced, they want the person to be in a coma and to be at rest, they might put a trach in, or for some other reason that they need help in ventilation. They actually have to section through the thyroid gland to get access to the tracheal cartilages to them. And you'll notice all the blood supply. So there's several thyroid arteries and even more veins. And that's because this is an endocrine organ. So it's very active metabolically, needs a lot of nutrients, and then a lot of blood supply carrying that material away to the venous system. And so when we get to the respiratory system, we're going to talk about how if you're going to do an emergency tracheotomy, you don't want to do it here, all right? Because of all that blood supply, all that blood flowing down into the trachea. So that's actually done up here. Okay, we'll talk about that when we get to the respiratory system. So the thyroid gland is controlled by hormones from the pituitary, which are controlled by hormones from the hypothalamus. Okay? So let's take a look at that pathway kind of as a review as we pull that back through. So we have the hypothalamus. And it's releasing thyrotropic or thyroid hormone releasing hormone. Now, this binds to cells in the anterior pituitary gland. Remember, this is getting from cap bed 1 to portal veins to cap bed 2. And so when this binds to cells in the pituitary gland, these are known as thyrotrophs. That's just what we call those clusters of cells that are in the pituitary gland. And this is a water-soluble molecule, so it's going to have a membrane-bound receptor. And this particular system is the phospholipase C. Okay? So when this binds, it's going to activate the G protein and the enzyme phospholipase C. And remember, that was the one with IP3 and diacylglycerol. So this is the activity inside the thyrotrophs of the thyroid gland that results then in the release of thyroid stimulating hormone. So we're talking about anterior pituitary gland here. Now that's going to get into which capillary bed? Capillary bed 2, and it's going to leave via portal veins, not portal veins, inferior hypophyseal veins, go to the heart, and then come back up through these thyroid arteries to get to the thyroid gland. Okay, so it's not going to go directly to the thyroid gland via the anterior pituitary. It has to get back to the heart first. And so it's going to go via I'll just put hypophyseal veins. To the heart, and then via thyroid arteries double A's is arteries. To the thyroid gland. Now, once it gets to the thyroid gland, It's going to go into smaller arterioles and blood vessels and capillaries and get to these cells that form the thyroid follicle, these simple tubular cells. All right, so I'm going to erase this sequence here, and I'll draw in the thyroid follicle. Yes.
So when the hypothalamus first releases its hormone, and of course it's sending it to the pituitary gland, like we learned yesterday, didn't it consider the pathways we had first learned, like with the, like you wrote, the phospho, I can't think of the whole name, phospholipase, yeah.
Yeah.
So, and that's on, or that's happening in the pituitary gland? Okay.
Yeah, because this is a hormone.
Yeah. Okay.
So it's binding to a hormone receptor, to the cells in the anterior pituitary.
Okay.
And most of the hormones from the hypothalamus use the phospholipase C system. We'll see next week as well. And so it's activating a G protein, which is activating phospholipase C, which is activating an inositol triphosphate, the GAG, and that activates the enzymes which regulate the synthesis of TSH.
So how do you know which pathway is the hormone is supposed to bind to?
It's been studied, so you know, because scientists have found those molecules in the cell.
Okay.
So it's, yeah.
So, but just putting it all together, so when the hypothalamus, the hormone left the hypothalamus, that's when you like said the portal system?
Right. So this would be, so in the hypothalamus, this is going to be cat bed 1.
Mm-hmm. And then it goes down the portal.
And then this would be the portal vein. And then it's going to leave via cat bed 2.
And be in the pituitary?
And go there. And then this is going to leave via cat bed 2 as well. So this is leaving, this is coming out of cat bed 2 and going to the pituitary. This is going into cat bed 2. Okay, so this is going into cat bed 2. This is coming out of cat bed 2. Okay, can I erase this now? Yeah, this room is kind of awkward, but all these boards, and I'm limited to this 2 feet by 4 feet.
So, I'm sorry, so you said that TRH is going into,
So TRH is coming
bed.
into cat bed 1,
okay,
traveling down the portal veins, leaving cat bed 2 to get to the anterior pituitary, and the resulting TSH enters cat bed 2, and then leaves the pituitary via pituitary veins. Okay?
thank you.
All right. Okay, now let's move to the thyroid gland. So in the thyroid gland, we have these spheres. Now they look like they're flat circular rings, but remember this is a section. So it's really a hollow ball, like a basketball, with a single layer of cells forming the wall of that basketball. The inside of the cell is filled with a liquid called a colloid. Right? And we'll talk about what makes up that liquid. But we're going to look at one of the cells of the follicles. Okay? It's going to have a basal surface, which is where the capillaries are available to it. And it's going to have a luminal surface, an apical surface, which is facing the follicle. Okay? So this is the lumen. Contains the colloid. We'll describe what the colloid is in a moment. And this is a simple cuboidal epithelial cell that has microvilli. So down here, this is the basal surface. And so the capillaries are going to be at the basal surface. Okay? Now, in our capillary is what molecule? remember what color. TSH, was that green? I think.
Yes.
So this is TSH coming from the anterior pituitary. It's also a water-soluble molecule, and it has receptors then on the basal surface. Size is not to scale, obviously. So these molecules are not going to pass into the cell. They're going to use the second messenger system, and this time it's the cyclic AMP system. All right, so the thyroid stimulating hormone binding to its receptor is going to turn on and activate the alpha G protein, which binds to adenylate cyclase and causes adenylate cyclase to change ATP to cyclic AMP. All right, so we'll just put cyclic AMP here. Cyclic AMP and the protein kinases that it activates is responsible for every single step that we're going to follow through. So it's going to activate enzymes that control each of the different steps that we're going to talk about with the synthesis of thyroid hormone. Even just that one molecule, a whole bunch of different enzymes that are involved in controlling this step. So, two major concepts are involved. Thyroid hormone, is that a glycoprotein, a lipid protein, modified amino acid? Anybody remember?
Amino acid.
Exactly, modified amino acid, and the modified amino acid is tyrosine. So, tyrosine is available to the cell from the capillary, but tyrosine is stabilized and bound to, several of them are bound to a long molecule known as thyroglobulin. So we have synthesis, modification of tyrosine and synthesis of thyroglobulin occurring. That's one major step that has to occur. But that modified tyrosine is not active without the presence of iodine on it. Okay? So the cell has to take up iodine, and convert it to iodine, and then the two are combined. The thyroglobulin, tyrosine, and iodine are combined within the cell. So those are the two sequences of activities that are going on. So I've got several different images. All right, some are more simple, some are more complicated. So I'll go through this image with you and then diagram it on the board. So here's our capillary. The TSH isn't shown here, but it's showing the iodine. Okay. Iodine is taken up into the cell. It's going against its gradient. All right, so there's more iodine inside the cell than there is outside the cell. So we have secondary active transport occurring. All right, well, yeah, this is important. We actually have a sodium channel, and sodium is going down its concentration gradient. Sodium is higher outside the cell than it is inside the cell, all right? And we maintain low sodium inside the cell by using active transport to pump it out. Well, that's where the active transport is going on. If you remember secondary active transport from Bio 430. So that maintains a low amount of sodium inside the cell. So this channel right here is open to sodium, and sodium enters by diffusion and drags iodide in with it. Alrighty? The presence of that pump is also due to cyclic AMP. Alright? So remember I said everything is going to use this cyclic AMP. So let's draw the pump in. I might have to enlarge my cell over here on the side to make room for everything. So we have a sodium, iodide, symport. Remember what symport means? Sodium potassium pump is an antiport. Sodium potassium going in opposite directions. So this is a sodium iodide symport. Sodium and iodide are both going into the cell. Iodide against its gradient but being dragged in because sodium is going down its gradient. So we have sodium going into the cell. and dragging with it iodine. So that's what's happening here. Then the iodide has to be added to our modified tyrosine, but that doesn't occur until up here. And it has to be converted to iodine. So we'll leave the sodium alone, just letting you know it gets pumped back out again by active transport. Okay. The iodide moves to the luminal surface, and there it moves through another channel. This kind of channel is called Pendrin. We'll talk about it again with the urinary system, but I'm not going to require that you know it here. It's a special channel for this type of ion. And on its way out, there's an enzyme embedded in the microvillus known as thyroid peroxidase. That enzyme is made active, guess what? By
Cyclic AMP.
cyclic AMP. Okay. So cyclic AMP activates protein kinases, which phosphorylates thyroid peroxidase and makes thyroid peroxidase active. What thyroid peroxidase does is changes this iodide to iodine, and it's drawn either as an I0 or an I+. So that's what happens with iodine. Any questions on that? Now, we now have to take care of tyrosine. Okay, so that's taking place within the ribosomal ER and the Golgi apparatus, and it synthesizes a protein known as thyroglobulin. So RAR to the Golgi, and we end up with a secretory vesicle containing a backbone of protein known as thyroglobulin. That's a string of amino acids. But sticking off of that string are individual tyrosine molecules. So this is the modified tyrosine. Okay. That's what's actually going to be our hormone. The thyroglobulin is more of a support. This is such a tiny molecule, it would move through the membrane and move other places, and so the thyroglobulin is our mechanism of keeping straight. So this thyroglobulin is released from the plasma membrane into the colloid via exocytosis. So the secretory vesicle fuses. Remember when we were in Bio 430, we talked about how vesicles would come to the surface, same thing with neurons, and fuse with the surface, open up, and then the content diffuses out. So this is what happens, is why it goes to the Golgi apparatus. So it has a destination, and in this case, the destination is the surface of the cell. And so it comes to the surface of the cell, fuses, opens up, and the thyroglobulin with its attached tyrosine molecules now is in the colloid. That's what makes up the colloid. So here's our thyroglobulin with its modified tyrosine. And once the iodide is in the iodine form, it will bind to the modified tyrosine. It doesn't need another enzyme to do that. Okay? So it starts binding to the tyrosine. Here's a video. I'm not going to show it now. It's about 7 minutes long. It's narrated, but I have it linked on D2L. I put it up there this morning, so if you want to go and watch it, it'll take you through some of these steps. So here's a couple of different views. So the orange string here, it's not in your diagram, the orange string here is the thyroglobulin. Here's the tyrosine. It's a single ring with a hydroxyl group on it. And here, 1 or 2 iodine molecules can be attached. Right? Then, these two can be attached to each other, and we have T4. If only one is attached, we would have T3. Okay? 90% of the thyroid hormone is T4. All of it's converted to T3 once it enters a cell where it's going to be active. All right? And that's the most active form. About 4 times more potent than T4 is.
So T4 is like a
It's kind of like the transport form of it.
transport form, okay.
All right, so there's a view of that. There's a more chemical, molecular view. All right, so thyroxine specifically is T4, and T3 is just triiodothyronine, but it can exist either as T3 or as T4. Okay. So this occurs, here's just another, I just put this picture up this morning. Here's another view of what we just got through talking about. So here's the sodium and iodine. I've got several different views, so whichever one makes sense to you. Here's the iodine coming in, going through its channel, and then the enzyme that converts it takes place right here on the membrane. Here's the synthesis of thyroglobulin with the attached tyrosine, and once it gets into the lumen, then the iodine is added to it. Right? So essentially what we find in the colloid then is thyroglobulin. Most of it will have the iodine added to it. Now this is very simple, but it has the steps on it. It just doesn't show it quite as clearly. So I actually like this diagram best as far as showing what's going on. Okay? So again, we have iodine. It doesn't show the sodium, potassium, and the sodium iodine symporter, but it comes through with the thyroid peroxidase enzyme here. It's converted to iodine, added as either 1 or 2 to the tyrosine, and now that's the storage form. So that's what we find in the lumen. Okay, thyroglobulin plus tyrosine plus 3 or 4 iodine molecules. Now, that's not the release form. So when the body needs thyroxine, when the body needs thyroid hormone, we have to endocytose this large molecule and deliver it to the bloodstream. But we have to get rid of the thyroglobulin. Okay? Thyroglobulin is a protein. So, can I erase this part here? Otherwise, I'm going to be going way over here. Not all of you will be able to see it on that end. Okay. So I'm going to leave this up here. I'll redraw it. So now, same cell, but it needs, the body needs to have thyroid hormone. So endocytosis occurs. This is internalized into the cell. So plasma membrane forms a secretory vesicle. And now it's inside the cell. And what do we do if we need to break down proteins? We need to break down the thyroglobulin and separate it from the modified tyrosine. Enzymes. And where are those enzymes found? In lysosomes. All right, so this endocytotic vesicle is going to fuse with a lysosome, which contains proteases. Those proteases will digest the thyroglobulin into individual amino acids and create free T3 and T4. The amino acids are recycled and used to make more thyroglobulin. So the T3 and T4 now are exocytosed into the capillary. So now this is going to be transported to the basal lumina. And released into the capillary. But you know what? It's not water soluble. So it is bound to a transport protein produced by the liver known as thyroid binding protein. That's how it's transported in the blood.
The thyroid binding protein will bind to T3 and T4?
Mm-hmm.
Okay.
So this is not, it passes through the lipid membrane. So guess how it works on a cell? Where would the thyroid receptors be for thyroid, for T3 to T4?
Inside yourself?
Inside the cell. Okay, so like we drew yesterday, once the T4 gets into the cell, it becomes T3. An enzyme removes one of those iodides. And so it's actually as T3, it binds to DNA, and just like our steroids, functions to cause DNA to synthesize messenger RNA and new proteins are produced. So just in this one pathway, we've gone over almost all of the different mechanisms, 3 of the 5 different mechanisms that are 6 that we use to talk about hormone pathways. We use the G protein, PIP2, with the TRH. We use cyclic AMP with TSH. And then with the thyroid hormone itself, the binding to the receptor within the cell. Okay. So thyroid functions, the thyroid hormone T3 in its active form, functions to increase cellular metabolism. We'll talk about this more when we do the digestive system. So it causes the mitochondria to make more ATP. So it increases your basal metabolic rate. Right? So individuals that have a high metabolic rate need more calories. So what would you expect, Narisis? What would you expect the symptoms of, and I think I have them a couple of them mixed up here, so let me double check. What would you expect the symptoms of hyper and hypothyroidism to be?
Hyper, unable to gain weight, and hypo, unable to lose it.
Okay, so let's start Narisis down. Okay, got it? All right. So if you have too much thyroid hormone, more than your body requires for basic metabolic needs, so hyperthyroidism, common sense, you're gonna have increased energy release. So you would have weight loss.